Okay, let's jump right into this explainer because honestly, as PhD level scientists and lab professionals, we have absolutely all been there. You're just sitting there staring at a completely failed cell-based potency assay, totally bewildered. I mean, it looked absolutely perfect during R&D, right? But now that it's in QC, the whole thing is just falling apart. So today, we're going to tackle the harsh lab reality of this exact problem. We'll talk about how to shift our mindset around troubleshooting and then take these concepts straight from theory right down to the bench.
Look, if you have ever frantically typed this exact phrase into a search bar out of pure frustration, usually after duplicate wells completely disagreed with each other, I want you to know you're in a safe space today. We have all done it. When an assay starts missing acceptance criteria, nobody wakes up and immediately thinks about, statistical philosophy. You just desperately want to know why your reference standards are suddenly behaving so erratically from one run to the next.
Well, the core issue we face at the bench really comes down to a fundamental difference. Predictable binding assays, they're basically like turning a key in a lock. It's just a single, clean equilibrium event. But cell-based methods, that is like orchestrating a chaotic living symphony. Unlike simple binding methods, you're relying on living biology here. And that means literally every single tiny change, from the cell passage number to the seeding density to the serum lot, is going to alter the measured response. The complexity is just way higher, which means the places where variability can hide are multiplied.
And when an assay is in distress, the symptoms are glaring. Honestly, they're kind of painful to look at on the screen. You might see high replicate CV, where your triplicate wells just wildly disagree, especially at those critical upper or lower response limits. Or maybe you're watching in horror as your EC50 drifts, with the fitted curve just sliding left or right day by day. Sometimes the dynamic range compresses entirely, so the top signal falls and the bottom signal rises. These failure patterns end up amplifying every weak control in your workflow, completely ruining your reference standards behavior.
So what do we usually do? We endlessly run the plate again, maybe changing just one minor thing, crossing our fingers for a pass the next day. We have to stop doing that. That guessing game just doesn't work. The curve drifting isn't the root cause at all. It's just a symptom. We really need to pivot our approach here and ask the tough question, what fundamental biological or execution variable actually shifted? And this brings us right to part 2, a massive, necessary mindset shift. If we actually want to solve this using true design of experiments, or DOE, we've got to start treating cells as variables, not constants. In way too many labs, quote-unquote bad cell health is just tossed around as a vague excuse for a failed run. But it isn't just a lab excuse. It's a glaring signal of a missing control strategy. Passage number, seeding interval, recovery period, these aren't just informal lab habits, they are critical method parameters. If they're drifting, you're not just measuring your sample anymore, you're measuring a moving biological system. To stop this endless guessing game, we need to instantly categorize our pain points before we change a single thing. You have to map the failure pattern. If your main problem is within plate noise, meaning replicates on the exact same plate disagree, that points squarely to execution timing, cell delivery, or local environmental effects. But if your plates look internally consistent, yet you're seeing between run drift across different days or different analysts, well, that points directly to biological state, cell preparation, or reagent lots. Timing drift is incredibly easy to underestimate. Just think about it for a second. An analyst gets distracted by a quick question in the hallway. Just 10 extra minutes between dispensing the first and last column can drastically alter stimulation time or attachment state or reporter expression. If you're doing manual additions and your biological response is fast, that simple 10-minute gap creates a devastating position effect that will absolutely ruin your CV. And this brings us to a huge culprit, the uncontrolled execution window. Listen, we have to stop blaming the curve fit model right out of the gate. Poor data analysis software rarely creates biological variability out of thin air. The real issue is that these informal lab habits, like having no guardrails for analyst handoffs, ignoring instrument cue to maze, sloppy wash timing, these are actually critical parameters driving your assay drift. High CV that clusters by plate region, yeah, that's an execution problem for sure, not a statistical one.
Okay, so how do we take this from theory straight to the bench? That's part 3. We've talked about the lab reality, we've covered the mindset shift, now we need a structured troubleshooting plan. How do we actually fix this systematically without running some massive, bloated, totally confusing experiment that just mixes up execution mistakes with biological unknowns? Well, before you commit to a huge redesign study, follow these 4 actionable steps. First, classify the failure pattern, just like we discussed earlier. Second, and this is key, reconstruct the true workflow. Write down what actually happened at the bench, not just what the SOP claims should happen. Track the true thaw date, the exact time from harvest to dispense, all those hidden pauses. Third, stabilize your controls. If your reference standards are unstable, tweaking sample dilutions is literally just wasted effort. And finally, run small targeted contrasts. Test 2 serum lots or 2 passage windows just to isolate the true bottlenecks before you launch into a full validation. Once you've narrowed down those primary suspects with their small contrasts, that is when we bring in DOE. Design of experiments is not some abstract statistical philosophy meant to just make a validation report look fancy. It's a highly practical, absolutely essential tool. You use DOE to map the interactions that are destroying your dynamic range. It gives you hard evidence that your method can actually survive normal day-to-day laboratory variation. By screening these specific parameters around realistic operating windows in a focused DOE, you build an assay that doesn't just magically work in one specific analyst's hands, but works consistently for everyone, every single time. Testing factors like cell seeding density, recovery time, and plate hold time all at once helps you answer which interactions are creating failures. Because sometimes, actually quite often, 2 conditions that are totally fine on their own are an absolute disaster when you combine them. And ultimately, this is exactly what quality teams and regulators care about. FDA and EMA guidelines all push in the exact same direction. Your assay has to be scientifically relevant and fit for purpose. Showing that your experimental design can completely separate that day-to-day analyst noise from true biological drift, that is exactly what proves your method can deliver consistent analytical conclusions across a real-world operating window. Which brings us right back to that endless guessing game we talked about at the beginning. The OFAT trap. Unstructured, one-factor-at-a-time reruns are the absolute enemy of assay transfer and long-term reproducibility. Blaming the cells and spending 2 months generating blind reruns might feel like you're doing work, but it just wastes time and completely hides the critical interactions between your variables. A structured troubleshooting plan is literally the difference between a one-time lucky rescue and a truly durable method. So, as we wrap up this explainer, I really want you to look closely at your current live cell potency method. Think about your workflows, think about your analysts and your controls, and I want you to ask yourself this highly provocative question. Are you actively controlling your biology, or is your biology secretly controlling your assay? Seriously, take a moment and let that sink in. And that brings us to the end. I really want to hear your thoughts on this. Drop a comment below with your biggest cell-based assay bottleneck right now so we can tackle it together in the community. And hey, if you found this structured breakdown helpful, please don't forget to like and subscribe for more deep dives into troubleshooting